If you've been studying the trip operon and finding it a bit confusing, finding it a bit hard to get your head around all the fine details and to even sort of work out actually what's going on, like for example, how does the ribosome get past that stop codon or the anti-attenuator loop that forms between regions 2 and 3 during attenuation? If those kinds of questions have been niggling at you and you haven't been able to get to the bottom of them, then I've made this video for you. Now, I do want to say that if you're a Unit 3 VCAA biology student, you probably don't really need all the detail that I'm going to go into in this lesson. As long as you've got a pretty good understanding of why the trip operon is necessary, as long as you understand repression and have a pretty good understanding of attenuation, then I think you'll be able to answer the questions that you find on the exam. But I do know that there are a lot of students out there who really do want to get to the bottom of it and who have these unanswered niggling questions that they just want to get their heads around. And I know that because I get emails daily from students and teachers too, for that matter, trying to make sense of it all. And there are so many questions in here that are actually the answers are quite elusive even when you search long and hard on the internet. Like for example, those structural genes in the trip operon, are they all translated by one ribosome together into a big long polypeptide that then gets cut up into 5 separate polypeptides? Or is each of those structural genes translated separately by a different ribosome? We'll answer those questions in this video. And I'll also, of course, explain the basic functionality of the trip operon as well. So if you don't care about all of that detail, if those questions hadn't even occurred to you and you really don't want to know the answers, that's okay. We'll cover the basics in here as well. Let's start by talking about tryptophan. It's one of the 20 amino acids that proteins are made of, and so that makes it pretty important in itself. Interestingly, tryptophan is the rarest of the 20 amino acids, and it's only encoded by one codon. You can see UGG encodes tryptophan. There's only one other amino acid that is encoded by a single codon, and that's methionine. All the others have redundancy, or they're degenerate. And what I mean by that, if you look at leucine here, for example, there are 6 different codons that encode the amino acid leucine. But that's not true for tryptophan. Tryptophan is also energetically expensive to make. It costs the cell a lot of energy to make tryptophan. And in fact, our cells can't make it at all. We need to get tryptophan in our diet. We call it one of the 9 essential amino acids. I've highlighted all the essential amino acids in this codon table. So we can't make it at all, but some bacteria can, like E. coli. And it's in E. coli that we find the trip operon. Because it's an energetically expensive, costs a lot of energy to make tryptophan, it's not in the interests of a cell to be making tryptophan unless it's needed. And remember, because it's a fairly rare amino acid, most proteins may only have 1 or 2 tryptophans. Out of hundreds of amino acids, they may only have just a very small number of those would be tryptophan. So because it's a fairly rare amino acid, it's not in the interest of the cell to be making lots of tryptophan until it's actually needed. That's the whole point of gene regulation, of the biosynthesis of tryptophan. There's more than one mechanism of gene regulation. In fact, there are dozens of them, and some of them are really, really complex. The reason we learn about the trip operon in Unit 3 biology is because it's a relatively simple example of gene regulation. Well, I mean, it's a sliding scale because there's nothing simple about the trip operon, but compared to some other mechanisms of gene regulation, it seems pretty simple. But the other reason why the trip operon is a really good operon to learn about is because by learning about the one operon, we get to see 2 very different mechanisms of gene regulation. So it's a good sampling of the sorts of ways in which genes can be regulated. So I like the trip operon. I think it's a really good example. First of all, let's think about what we mean by the trip operon. What actually is an operon? Well, an operon is something that we tend to associate with prokaryotes. There have actually been examples of operons now found in eukaryotes, in Drosophila flies, for example. But typically in eukaryotes, every gene has its own promoter. the place at which the RNA polymerase assembles to transcribe the template strand of the DNA. But in prokaryotes, it's quite common for a bunch of genes, often genes that are related in function, that are all involved in the same biochemical process, to share a promoter. And that's exactly what we find in the TRP operon. There are 5 structural genes that we call TRPE, TRPD, TRPC, TRPB, TRPA. Notice that it's A, B, C, D, but they're in the wrong order. But anyway, that aside, these 5 structural genes all share one promoter. And what that means is that they're all transcribed together or they're not transcribed at all. That is what an operon is. A set of structural genes that share a promoter, so they're all transcribed as a package deal. All right, so let's have a look at what happens during the first mechanism of gene regulation, which we call repression. But before we even get to that, let's just look at what the operon does. What's the purpose of this operon, and what are these structural genes? What actually happens? All right, well, as I said, RNA polymerase assembles at the promoter, and then you'll notice that between the promoter and the structural genes, there's an operator and a leader. Well, there's another difference between prokaryote genes and eukaryote genes, is that prokaryotes have this region called an operator after the promoter. In eukaryotes, we don't have an operator. We have other things, other mechanisms that achieve the same purpose, but we don't have this operator between the promoter and the gene, the structural gene. Anyway, what will happen here in the TRP operon is RNA polymerase will assemble at the promoter just like it does in any gene, and then it will run past the operator and the leader and transcribe a messenger RNA molecule, which is a copy of the complementary strand of the leader and the structural genes. It uses the template strand to make a copy of the complementary strand. Now, there's a couple of, I think it's worth pointing out some other differences between prokaryotes and eukaryotes in this regard, in that prokaryotes have no introns. So RNA splice, in the eukaryotic genes, after transcription, the pre-messenger RNA has to be modified in a process we call RNA processing. Introns get cut out, a methyl cap gets added to the 5' end and a poly-A tail gets added to the 3' end, right? That's what happens in eukaryotes. But in prokaryotes, there are no introns to cut out, and a poly-A tail doesn't get added to the 3' end, and a methyl cap doesn't get added to the 5' end either. Instead, there are these little regions that a ribosome will be able to attach to in order for translation to take place, instead of needing that methyl cap at the 5' end. All right, so just again, another difference between prokaryotes and eukaryotes: no introns, no methyl cap, no poly-A tail. So as soon as transcription is finished, we have a mature messenger RNA molecule. That messenger RNA molecule then is translated as happens in eukaryotes, and that of course happens as a result of the action of ribosomes, giving us a polypeptide for each of those structural genes. Now, the first 2 of those polypeptides, the ones that are transcribed and then translated from TRPE and TRPD, they together form an enzyme called anthranilate synthase. It's an enzyme that has quaternary structure. It's got more than one polypeptide. The polypeptide transcribed and translated from TRPC becomes an enzyme itself, which takes anthranilate, which is the product of anthranilate synthase. It takes that anthranilate and converts it into indole 3 glycerol phosphate. And so, of course, this enzyme is very appropriately named indole 3 glycerol phosphate synthase, because it synthesizes indole 3 glycerol phosphate from anthranilate. And the last 2 polypeptides from TRPB and TRPA, they form another enzyme, again with quaternary structure, called tryptophan synthase. And tryptophan synthase takes the indole 3 glycerol phosphate and converts it into tryptophan. So that's the biochemical pathway that results in the production of tryptophan. Now, like we said earlier, that's very energetically expensive. It costs a bacteria a lot of energy to be able to make that tryptophan molecule. So we don't really want that to happen unless it's really necessary. And that brings us to gene regulation. The first mechanism of gene regulation that we find in the TRP operon is called repression. And repression mechanisms either turn a gene on or turn a gene off. They repress them or they're unrepressed, on or off. And it happens by way of this protein called a repressor. And the repressor is a protein that's encoded by a gene elsewhere in the bacterial chromosome, quite a long distance away from here. But nevertheless, you can see in this cell at the moment, there's heaps of tryptophan. Look at all that tryptophan, stacks of it. There's also charismate, of course. There's always charismate in cells. Charismate, dime a dozen, it's all over the place in cells. Tryptophan, remember, is generally pretty rare, but in this cell, there's plenty of tryptophan. So it would be a real waste of energy for this cell to be making more tryptophan. So what happens is that tryptophan binds to the repressor, and like often happens in biology, when molecules bind to each other, particularly to proteins, very often those proteins have a conformational change, or they change shape. And when that happens, the repressor now has the perfect shape to fit onto a site on the operator. And what that does is it blocks the operator so that RNA polymerase is blocked from transcribing the leader and the structural genes. Okay, so transcription doesn't happen. It's been turned off or repressed. Of course, once the level of tryptophan goes down in the cell, if it all gets used up, then even the tryptophans that are stuck in that repressor will come away. The repressor would go back to its regular shape, come away from the operator, and then the RNA polymerase can transcribe the operon as it normally would. Okay, so it's temporary. In fact, that repressor sort of jumps on and off, back on and off. It doesn't stay stuck on there like glue. It actually falls off quite frequently and goes back on again. So even when there's tryptophan in the cell, a low level of tryptophan will still be able to be being made because, as I said, the repressor doesn't stick on there hard. It sort of sticks on there and then it falls off and then it sticks back on and falls back off. So it stops the RNA polymerase, but every now and then an RNA polymerase will get through and a little bit of tryptophan will be synthesized anyway, even when there's tryptophan available in the cell. All right, so that's how repression works. I think repression is relatively simple to get your head around. It's attenuation that confuses most people. This is the second mechanism of gene regulation in the TRP operon. All right, now to understand this, we need to look at the leader region. Remember, this is the region between the operator. So to the left of the screen would be the promoter. To the right of your screen, you see the structural genes. You can see the start of TRPE, which is the first of those structural genes. And I've colored this orange bit here, orange and brown, the leader region. Now, there are a few things that we need to notice here in the leader region to make sense of attenuation. If you don't understand all these bits, if you don't know all the bits that I'm about to explain, then it's very hard to see how attenuation can work. So firstly, notice that there's a big difference between transcription happening in or translation rather, happening in both actually, happening in a prokaryote and transcription and translation happening in a eukaryote. In a eukaryote, there's a nucleus. So transcription happens in the nucleus and then the messenger RNA leaves the nucleus, goes out into the cytoplasm where it's translated by a ribosome. But in a bacteria, there is no nucleus. There's no nucleus. So there's no nucleus for the messenger RNA to go out of. And because of that, there are ribosomes mixed up in here with the DNA. So as soon as transcription happens, even while transcription is happening, the ribosome will start translating the messenger RNA. And you can see that's exactly what's happening here. We've got the RNA polymerase running along here, taking the template strand of DNA, using the template strand of DNA to make a messenger RNA copy, which is what we're looking at right here. But even while the RNA polymerase is transcribing, a ribosome has jumped on here and is translating at the same time, following it along, the two of them following along in tandem, one transcribing, the other translating hot on its heels. All right, so that's the first thing to understand. The whole idea of attenuation wouldn't work if that weren't the case. Attenuation wouldn't work unless translation was happening at the same time as transcription. That's the first thing to get in your head. Secondly, notice that there are these 4 regions that I've called 1, 2, 3, and 4. Well, I didn't call them 1, 2, 3, and 4. That's what they're called. Region 1, Region 2, Region 3, and Region 4. Now, they are important, very important, because they're complementary to each other. And I'll show you what that means, what that looks like in just a moment in more detail. But essentially what it means is there are parts of Region 1 that are complementary to Region 2. There are parts of Region 2 that are complementary to Region 3, and parts of Region 3 that are complementary to Region 4. Now, what that means, like let's say that there was perhaps a CCC here, there might be a GGG here, and because CCC is complementary to GGG, they could theoretically stick to each other. Same with 3 and 4. There might be perhaps UUU here and AAA here, and because U and A are complementary to each other, they could theoretically stick to each other. Okay, so 1 is complementary to 2, 2 is complementary to 3, 3 is complementary to 4. Also, I want you to notice that there are 2 codons for tryptophan. 2 of that UGG codon that specifies tryptophan right here in region 1. And there's a stop codon between region 1 and region 2. They're absolutely important. Okay, so a double codon right next to each other. Okay, UGG, UGG is how it reads. And then a stop codon between region 1 and region 2. Okay, so all of this we call the leader. It's also, I think, interesting that from here to here, this ribosome actually makes a little polypeptide. It's not, we don't call it a polypeptide, we just call it a peptide. It makes a little peptide of 14 amino acids. You know, that becomes important later. All right, so it is actually translating and joining amino acids together here, but it's not along here. This is just kind of meaningless gibberish. It's not used to make anything, but up here, of course, the structural genes do. All right, let's move on. What we're looking at here is the leader region. You can see here's the operator upstream, and you can see there's the TRPE gene, the first of the structural genes downstream. So it's kind of like what we were looking at in the last slide, but just now we're actually looking at the nucleotide sequence, the base sequence that is in the leader region. And remember I said that to begin with, there are 14 amino acids, a peptide containing 14 amino acids that's translated from the start of this leader region. And then there's this stop codon, right? So there's that stop codon that we talked about. But notice that here's our 2 tryptophan codons, UGG, UGG. So again, you know, that's exactly what we were looking at in that previous slide. UGG, UGG, tryptophan, tryptophan, and within region 1, and then between region 1 and region 2, there's a stop codon. Okay, so this is exactly what it looks like. But notice in here, I've shown how region 1 and region 2 are partially complementary to each other. So they can sort of bend over and stick to each other because, you know, A is complementary to U and G is complementary to C and so on. And the same thing can happen over here between region 4 and region 3. There's this region there, this stretch, where there's a whole bunch of cytosines that are complementary to the guanines in region 4, and so they will form hydrogen bonds between them to form this kind of a hairpin loop right there. One thing that I didn't point out in the previous slide, but I want to point out now, is that following region 4, there's a region called an attenuator. And that's another important thing to understand in order to understand how attenuation works. Obviously, it's a pretty important thing because it's called attenuator, and this is attenuation, so it's named after the process. So obviously, this is a pretty important part. But notice the attenuator is full of uracil. The messenger RNA attenuator is full of uracil. Obviously, it's complementary to adenine. The thing to know about adenine and uracil is that they only form 2 hydrogen bonds between them, whereas guanine and cytosine, like this pair here, form 3 hydrogen bonds between them. That makes it a stronger bond, right? If you've only got 2 hydrogen bonds, it's a weaker bond. You know, they're held together less strongly than if there are 3 hydrogen bonds. Cytosine and guanine stick together hard. adenine and thymine stick together only 2/3 as hard. All right, good. So that said, I also want to point out, this is exactly the same as the previous picture, except that notice it's also possible for region 2 to pair with region 3. In the previous slide, we had region 1 pairing with 2 and 3 pairing with 4. But in this slide, we've got region 2 pairing with 3, because again, notice that there are parts of it, a large part of region 2 is complementary to a large part of region 3. So they can stick together too, and again, form this hairpin loop like that. Okay, but we're still looking, there's our operator, there's the 14 amino acids, still the same, there's the last 3 of those. Then we have our stop codon between region 1 and region 2. So it's exactly the same thing. We're just looking at it, because remember, this is a molecule, so it can move around, snake around all over the place, and one part of it can stick to another part very easily. And so I've just shown it in two different conformations, 2 different ways that it could pair with itself. So that is really important. Now, with all of that background, we're ready to understand how attenuation takes place. Okay, so here we have what happens when there's still some tryptophan available in the cell. Now, there's no free tryptophan. This is what would tend to happen when most of the tryptophan just floating around in the cytoplasm is gone. Okay, there's not much free tryptophan, tryptophan, that's easy for me to say. There's not much free tryptophan just floating around in the cell, but there is still tryptophan being held by transfer RNA molecules. They're the molecules that bring amino acids to the ribosome for the purposes of translation. Now, because there's plenty of transfer RNA molecules with the anticodon ACC, that would pair with UGG, and bring tryptophan up to the ribosome, you can see that as the ribosome has just run past this spot where there are 2 codons specifying tryptophan, 2 transfer RNA molecules have just inserted a tryptophan there, representing those 2 tryptophan codons. Okay, so the ribosome has run past there, stuck a couple of tryptophans in, but when it gets up to here, it hits a stop codon and stops. That's what a stop codon does. It gets to there and it will stop. And because it stops right there, it's partially overlapping region 1 and partially overlapping region 2. And so it kind of prevents region 1 from bending around and reaching region 2, or region 2 bending around and reaching 1. And it also prevents region 2 folding around and sticking onto region 3. Because it's kind of holding them. It's like it's grabbed them by their shoulders each and is keeping them in a straight line, so this can't bend to that. So meanwhile, while the ribosome is stuck here waiting, doing nothing, just stopped at the stop codon, the RNA polymerase continues along, transcribing. And so it goes all the way along here past region 4 and into this attenuator region where there's all these, adenines and thymines, which were now uracils and adenines, I should say, because it's the messenger RNA here. So because the RNA polymerase has run along there and transcribed uracil, 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 but it's gone past region 4. Region 4 is attracted to region 3. So it pulls towards region 3. And that pulling, as it pulls towards region 3, it yanks the messenger RNA away from the template strand of the DNA. Okay? The tension as it pulls this way yanks it away from that messenger RNA. There's enough tension to pull it away. Now, that wouldn't happen if there was a whole lot of cytosine and guanine in here, because they would be stuck together more with more hydrogen bonds. But remember, adenine and uracil are weaker because they've only got 2 hydrogen bonds, and weak enough that the attraction between all the cytosines and guanines in here will pull this area of uracil away from this area of adenine here. And when that happens, the RNA polymerase flies off. It sort of, as the messenger RNA gets rent from its grasp, the RNA polymerase falls off the DNA and transcription stops. And that's a good thing. That it's been attenuated. Transcription has been attenuated, slowed down, prevented. And the reason that that's a good thing is because there's still tryptophan in the cell. Sure, there's no free tryptophan floating around, but there's tryptophan there nonetheless, attached to transfer RNA molecules. So even though maybe the level of tryptophan is getting low in the cell, there's still some, and it's too early yet to start spending energy to make new tryptophan. We don't need to spend energy until we absolutely have to. We don't have to yet. And this mechanism prevents that from happening by attenuating transcription. Now let's have a look at what happens in the cell when the tryptophan is all but gone. Now, there'd be still some tryptophan somewhere in the cell floating around, but most of it is gone. You can see that even our transfer RNA molecules that would normally carry tryptophan are uncharged. They've got no tryptophan on them. So now it's time to start making tryptophan. Now it's worth spending the energy to make the tryptophan so that the cell can make all the proteins that it needs to make that contain tryptophan. And you can see that what's happened here is that the ribosome has started translating this leader region, these 14 amino acids that form this peptide from the leader region. But when it gets up to the tryptophan codon, UGG, it just sits there waiting for tryptophan. It's not like it continues making the polypeptide and just leaves a gap in it or something. It just sits there and says, well, I'm waiting, and it will just sit there waiting until finally a tryptophan is delivered to it by a transfer RNA. Meanwhile, though, it doesn't get up to the stop codon because it stopped earlier. It stopped earlier than the stop codon. And remember, when it got to the stop codon, it covers up part of region 2, preventing region 2 from bending around and sticking onto region 3. But now that the ribosome's not blocking region 2, there's nothing stopping region 2 and region 3 from sticking to each other, and they do because they're attracted to each other because all those complementary cytosines and guanines that they pair to each other, and so 3 and 2 pull together. But it's a little bit different this time because the ribosome hasn't yet made it up to here. Okay, and so as a result, because this has happened, but it happens earlier because the ribosomes made it past 2 and 3, but it hasn't yet got up to 4. Earlier what had happened was that it had gone on longer because the ribosome had gone all the way up to the stop codon. Meanwhile, the RNA polymerase had traveled further along because they're traveling along at the same pace along the DNA. So they'd both moved further along. The ribosome hasn't moved as far. The polymerase hasn't moved as far either. And so as a result, when the ribosome stops, the polymerase hasn't got up to the attenuator region yet. And so this pulling, this tension between regions 2 and 3 doesn't pull the messenger RNA away from the template strand of the DNA because this is region 4 here. And there's a lot of cytosine and guanine in there. It's held together much stronger than the attenuator region would be with all of its uracil and adenine. So the tension here is not enough to pull the messenger RNA away from the DNA. And as a result, the RNA polymerase doesn't fall off, and it continues along here and translates all of the structural genes into the polypeptides needed for the biosynthesis of tryptophan. Well, that's all great, of course, but it does raise some interesting questions, and these are those niggling questions, those annoying questions that some of you have been plagued with, where you just can't find the answer to. So let's have a look at those now. One of the questions that people ask is, how can the ribosome get, because here we are in a situation where we need to make more tryptophan, so clearly these genes, these structural genes, need to be translated. But how does the ribosome get past this anti-attenuator loop, right? How's it going to get past this kink in the messenger RNA? Well, there are two parts to this answer. The first part is that ribosomes are able to smooth out these hairpin bends in RNA. And this is not the only place. RNA will often stick onto itself because it has little bits of complementary regions all over the place. And ribosomes move along the RNA with enough force, 13 piconewtons if you care, which is more than enough force to smooth out a bend like that in the messenger RNA. The second part of the question, though, is it doesn't matter. It doesn't have to get past the hairpin. Another question that people ask is, well, how can the ribosome get past the stop codon? And again, there are all sorts of answers that you might have read on the internet, like that some ribosomes, some stop codons are leaky and ribosomes are able to get past them, particularly if protein synthesis is low, and there's all sorts of complicated things. That's the first part of it, is that sometimes ribosomes can get past the stop codon under strange situations, but the second part of the answer is that it doesn't matter. The ribosome doesn't have to get past the stop codon. And the third question that people often ask me is, how does it get past, how does it get unstuck from waiting here for a tryptophan? Well, again, this is less of a problem because there's going to be tryptophan in the cell somewhere. It might not be readily available. It might have to wait a while, but eventually some tryptophan surely is going to find its way here. So that's not really a problem. But the second part of the answer to that question is that, well, it doesn't really matter. The ribosome doesn't have to get past these tryptophan codons. Why doesn't it matter? I hear you asking. You keep saying it doesn't matter, it doesn't matter. I'll tell you why it doesn't matter. It doesn't matter because this ribosome is not the same ribosome that translates the structural genes. And I can show you why we know that. Here we are looking at the first two structural genes. Well, it's the whole of TRPE and the first half of TRPD. Now notice, this is like any gene, starts with AUG. All genes start with the start codon, starts with methionine, and it keeps on, the ribosome will start translating here, start adding amino acids for each of these codons until it gets down to the stop codon UGA. At that point, it will leave the messenger RNA. Okay, so in other words, each of the structural genes is translated by a different ribosome, or it might be the same ribosome that leaves and then comes back and joins on. Who knows? It might be actually the same physical thing, but they're each translated in a separate translation event by a ribosome. Okay, and how do we know that? Well, we know it because we can look for ribosome attachment sites. Now, in eukaryotes, this is different because in eukaryotes, ribosomes attach where the methyl cap is at the front of the messenger RNA. But in prokaryotes, ribosomes attach to these sequences that contain adenine and guanine called ribosome binding sites, RBS for short. So this is one of the ribosome binding sites here. There's a bunch of them, but they all have adenine and guanine in them, different amounts and whatever. But GAG is a ribosome binding site. So when a ribosome sees that, it will bind onto that and start running along the messenger RNA, and then it starts translating when it gets here, goes all the way down to here where it stops. Meanwhile, notice that TRPD starts with AUG here, but the ribosome binding site for it is here within the TRPE gene. So a ribosome will bind to the TRPE, the end here of the TRPE gene, and run along here until it hits AUG and then starts translating these codons into an amino acid chain. So isn't that fantastic? There's actually overlap between the structural genes, and each of the structural genes is, as I said, translated separately into a polypeptide in a separate translation event, probably by a different ribosome. Let's have a look at how that looks back on the diagram we were looking at before. You've seen this diagram before, most of it anyway, but here we are. This is the ribosome that binds upstream of the leader region. Okay, so it binds upstream, and notice there's a ribosome binding site with adenine and guanine in it. It binds there, it runs along there, starts translating, that's the 14 amino acids in the peptide from the leader region, and then it stops. And the ribosome leaves the messenger RNA at that point. It doesn't need to get past the stop codon, and it doesn't need to get past this hairpin because there's another ribosome binding site here before the TRPE gene begins. And so another ribosome will just latch on there and start running along here, translating the TRPE gene. And that will be the case for all of those structural genes, some of which overlap with others. It's really wonderful, isn't it? What a magnificent piece of biology to understand, this operon. I really hope that that's helped you to understand some of the things that you were struggling with, and that it all makes sense to you. And if it does that, then the video's achieved its purpose.